There are differences between axi-cel, T-cell, and liso-cel, and you really have to go down to the CAR T-cell construct itself. While they all are targeting against CD19, so that receptor that lives on the outside of the lymphoma cell or the diffuse large B-cell lymphoma cell, what then happens after engagement may be a little bit different. And that is represented in the costimulatory molecule. So in the axi-cel construct, that is CD28, which provides signal 2, whereas in T-cell and liso-cel, 4-1BB is the costimulatory molecule. I think the other differences that has to be called out between the constructs is the dosing. So the dosing with axi-cel is given on a weight-based formulation up to a total cell dose of 2 × 10⁸ CAR T-cell cells, whereas T-cell is actually a variable dosing. It can go anywhere from 0.6 × 10⁸ all the way up to 6 × 10⁸ CAR T cells. And then lastly, with liso-cel, that CAR T construct is actually split between a flat dose of CD4 cells and a flat dose of CD8 cells, so split dosing. But currently, I believe in the core data set, the total cell dose is 1 × 10⁸ CAR T cell doses.
It's hard to compare actually the 3 studies, partly because they weren't done as a phase 3 randomized study. And as I said, there were some differences in patient populations. I would say that there are major differences in the product themselves, which I think is probably the most important thing. We know that there are, in this sort of second generation of CARs, it became apparent that one needed to have a so-called co-stimulatory molecule to get expansion of these cells. Without the co-stimulatory molecule, expansion of these cells is very difficult. And there are 2 co-stimulatory molecules that are currently in play, and those are the CD28 molecule, which is what's used in the ZUMA studies, and something called a 4-1BB co-stimulatory molecule, which was used in the Transcend study from Juno and in the Juliet study from Novartis. And the other difference is in the Transcend study, there are data from preclinical data from Stan Riddell at the Hutch in Seattle that suggested that an equal amount of CD4 and CD8 cells infused in an animal model might be beneficial in terms of efficacy. And so in the Transcend study, there's a fixed ratio of CD4 to CD8 cells that go into the patient, whereas that ratio isn't pre-established in the ZUMA 1 study or in the Juliet study. Now, whether that makes a difference or not, we don't know. I think certainly the preclinical model suggests that it will, but we really have no evidence that in patients it's going to make a difference. In fact, I think when the Kite group looked at their ratios kind of retrospectively, they were for the most part, I think, close to 1-to-1. So although it wasn't necessarily planned, the ratio turns out to be 1-to-1. So whether that's going to make a difference or not, we don't know. There was also some difference in dosing in the studies. The doses really weren't the same, perhaps a little bit higher doses in the ZUMA 1 study. So I think it's hard to compare across studies. There is a sense that CD28 as a co-stimulatory molecule leads to more rapid expansion of the CARs, whereas the 4-1BB co-stimulatory molecule leads to a less rapid expansion and perhaps more sustained availability of those cells. So I think all of this is going to have to be sorted out, and whether there will actually be a randomized comparison ever, I don't know. I think that's going to be a difficult trial to do, and whether that's the most important question right now, I can't say.